combinations of things going on, different recombination events going on, that we also do account for with the number of SNPs that we want to use. But in this case, we want an R squared to be above 0.8. So all this slide is telling you, these are different microarrays technologies that use certain numbers of SNPs. And what it's saying is that for our data in particular, if we want to get 0.8 or above, this chip does that 66% of the time, this one does as well, these do it a lot lower. So if I wanted to use a chip based on my data, I'd want to use one with the highest percentage of 0.8 or above. So whatever the S&P array is 6.0. Because in that case, we're getting the highest level of R squared that we can get on that chip. And it's still in 66%, but for this point, it's good enough. So I would assume this chip may be more expensive. It's using more SNPs for us to ensure that we get the nice R squared value. But once again, if you're looking at very small, if you have a very small sample size, you need more SNPs to get a higher R squared value. If you're looking at a disease that only 10 people in the world have it, you got to use a hell of a lot of SNPs. If you're looking at something like type 2 diabetes, that a lot of people have, you can lower the number of SNPs you need to still get a high R squared value. You can go here, look at this GWAS Central, and sort of understand this going through. So that's this lecture. A lot of technology stuff, some stat stuff and how it's related to it. So this is sort of like the nitty-gritty of how we've done everything up to this point for how we determined the disease, this gene associated with it. So if we come with this, blank likes all this stuff a lot, unfortunately. So the only thing likes more than this is the cancer genome stuff. Coming up next. What's up?
I had a question on the recombination between the DR and DQ paper. Was the significance of that was just that there was recombination in that halotype block?
Yeah, so the significance is almost always there shouldn't be.
Yeah.
And so, that's why we have the DQ and DR always going together from parent to offspring, because the mom had it, or the mom's chromosomes and it went through. In the case of that was a unique example where we did find recombination within it. So, yeah, it's reminding us that this isn't all or nothing. It's a high frequency or low frequency that will occur.
But then did it have any, because that paper was talking about type 1 diabetes, did it have any effect on the disease state within the child?
Yeah, so I believe, and this is a good point I forgot to mention. So we have good and bad things with haplotype blocks. If I identify the block and I know that all the disease patients have that block, let's say they all have this one right here. This DR, I don't know if this is what it is. Let's say everyone that has this block has the disease. What that tells me is one of those genes is screwed up. But do I know for sure which one it is? Which of those DRs or DQs? That's one of the limits of using haplotype blocks. I can never for sure know, oh, it's this gene within the block, not that gene within the block. The only way we can get more info on that is if we do get a very rare chance of a recombination occurring. So if, for example, this person, this is their block, if they still have the disease, that then tells me then, if this is the main disease, this is the haplotype, then it has to be one of these first ones. It's this first DRB1 or the DQA1. Does that make sense?
Yeah.
So yeah, it's a way for us, once again, to get, it's like that first picture where we had like 8, 3, 2, and then there was the orange, and then it was the other one recombined. I know for sure it's somewhere in there.
Okay.
So I know for sure it's one of those DR or DQs from the beginning.
And then what's like the typical size of a halotype block, or is it like variable, or?
I think it varies, but I think it's like on a kilobase scale, so it's like it's big. They're big. They're not very, very small. All right. Any other questions before we get to TCGA? Okay. So. You are going to have questions on the exam that are going to be like, this data set versus this data set, is it unprocessed or processed data? You need to know it well. I don't know it that well. I did it well in the first 2 exams, so I kind of like looked at this the more I know of the exam. I'm going to explain it the best I can, but I'm still not very comfortable with some of the numbers. Like, sometimes I feel like there's conflicting information he gives us, and it confused me. So I'll try to look at that and hopefully you guys can answer. If you guys know it, definitely help me out. But the whole idea here is we're looking at the cancer genome, so it's going to be looking at TCGA, CBioPortal, But aside from that, we're looking at the other thing, which is unprocessed data. Which literally, the key thing for me with unprocessed data, it's literally the reads of the DNA. So it's literally all of the DNA, just all of the sequences. And you get that. You can't just go online and get this. You have to get requests from where? NIH. So yeah, I know that's like random, but Blank sometimes likes those little things. So it's the NIH you go to, and you have to request these reads. And they literally then just give you thousands and thousands and thousands and thousands of the reads. And then you have to work with that. The cBioPortal, that's processed data. Someone else took that data and then look at it how they wanted and then put all the results in there. That's like looking at people with, looking at melanoma with BRAF mutation, things like that. So the one good thing with the processed data is a little easier to deal with. It's like not all the reads. You can understand it really better. The bad thing with the processed data, someone already processed it. So I can't look for things I want to look at in the DNA. If it's unprocessed, I can write my own code and look for certain variations that I want to look at. If it's processed, you can't work backwards from there. So both of them have their benefits. Sort of keep that in mind as we go forward. All of these studies, Blank is the main author on them. All of them. So make sure you know them fairly well. So this first one basically is looking at big mutation targets, and then but they're all dealing with cancer. Obviously, it's the cancer thing. So let's just go to the nitty-gritty. It's this slide and the next slide. Basically, what Blank did is they looked at all these different cancer types and what happens with cancer in general. What causes cancer? It's due to mutations, right? And typically, what do we lose first? What are the first things we're going to mutate? Usually. Checkpoints, DNA repair. So once I lose DNA repair, I can't repair mutations, then mutations build up. So here, they're looking at, in different cancer types, how many mutations were there. And not just that, I want to look at total mutations, and I want to compare it to actual important mutations, so like ones that are going to cause changes to oncoproteins or tumor suppressor proteins. A lot of these mutations will be silent mutations. What does that mean? I'm changing the base pair, but I have no effect on the amino acid. So they're basically looking at mutations in general, but comparing that mutation level to mutations that only dealt with changes to oncogenes, oncoproteins. Or basically those driver mutations, so the ones that are the important mutations that lead to this phenotypic effect. And basically and that's basically this slide showing us here, and then this over here, they're looking at, for each of these different cancer types, they put people in different groups. High frequency mutation, low frequency mutation. Which means that some people with bladder cancer, those cancer cells, one group, there were a shit ton of mutations across the board. A lot of oncoprotein mutations and a lot of silent mutations. So mutations that didn't have an effect. Versus another group didn't have a lot of mutations. They still have the bladder cancer, however, they had a lot fewer overall mutations in general. So they took all that data and then they compared it to each other. And they basically, once again, we're keeping it in different groups, but they basically wanted to look at, and the main question they were asking is there a statistical difference in different cancer types between having a high number of overall mutations or a low number of overall mutations? Keep in mind, what are the two data populations they're looking at? It's not disease and normal, it's just disease, but disease with a bunch of mutations, disease with fewer mutations. What they found in two of the cancer types, colon and lung cancer, is that there was a really big difference where, excuse me, in terms of the overall amount of oncoproteins relative to the total mutations that occurred. So, bringing it back to what we talked about with this in particular, this study, sometimes you got a bunch of mutations, but only a few of those mutations were oncoproteins that got mutated. So you have a bunch of mutations on the bottom, denominator, numerator is oncoprotein mutations, but there weren't that many of them relative to the total number of mutations. In these two groups, the colon and the lung, we had fewer mutations overall, but of those mutations, more of them counted. More of them were mutations in the oncoprotein. That should kind of make sense. Because if I tell you you've got bladder cancer, but you don't have very many mutations, where are those mutations are going to be occurring? And the things that are going to be responsible for causing the cancer. So a lot of those oncoproteins. Or if I say you've got bladder cancer, but a bunch of mutations, a lot of them occurred, but they're silent, or they don't really affect the phenotype. So that kind of make, a lot of these studies, they're kind of like, it's nothing revolutionary, but it's going to be, they're just looking at things that we would expect, but they're confirming that for future research and things like that. So once again, that's why they have very low p-values. Lower p-value is better. That's talking about there is a much greater incidence of those oncoproteins relative to the total mutations we see in that cancer type for these data sets. Once again, processed, unprocessed data. Well, yeah, what's up?
Could you go back to the last slide? I'm kind of confused. Like he's saying that relatively you have more oncoproteins mutated compared to all mutated genes. But I mean, according to that, it looks like glioblastoma relatively would have more oncoproteins compared to all mutated genes.
The key thing to look at is here. So this is saying that this is 3.8 oncoproteins over whatever the total all mutations is going to be. So it's the relative ratio of each. So even though I have a high amount of oncoproteins mutated, that bottom number is total of that cancer type. So that's why in that case, this is the really important value, is this is the ratio of onco to normal.
Oh, so
yes.
that is a ratio right there.
Yeah, it's the ratio of total onco versus all the mutations we had there, and only in the colon group and the lung group for those cancer types do we have a very high ratio, which means that when we got those mutations in those cancer types, they really counted. Because there were some other ones that were silent, but these other groups had a bunch of silent ones typically. Yeah, good question. Processed, unprocessed data. So key thing here, processed data, no normal tissue. So we do not, and this is one of the things where I think later, when we get there, I'll bring it back up, but you need to be aware of examples of each of these. He's going to throw that on the exam. He's going to say, this is the data set, is it processed or unprocessed? It's going to be A or B. And hopefully it's one that we're comfortable with and we can answer correctly. But the whole idea here, once again, is that it's going to be biopsies of the tumor, but we're only looking at the DNA of the disease state. There is very little to no comparison to normal tissue DNA, or normal tissue of the patient. And then we're also going to have things like Excel files and text files. But big thing here is cBioportal. That's the big one. Versus unprocessed. It's literally the reads right off the machine. So the Illumina reads of all of that sequencing, it's all just, remember like that long, you know that example he gave you guys for second exam? It was like 5 pages of the reads. That's literally the unprocessed data. That's what you have. That's all they give you. And you then have to write a code for it to see what the hell they're going to look at. In that case, this is available through Genome Data Commons. Once again, you ask for the ability to use it through NIH, and then it's all through the Genome Data Commons. That's the equivalent of cBioportal. This is where you have all of the unprocessed data for all these patients. You ask for those reads, and then you're able to go from there. So we'll start with the processed data. And this is why it's confusing, because he puts transcriptome and then whole genome methylation, but he just said it's not the whole genome, it's just a disease state, so I'm a little iffy. He has transcriptome here, but then when we go to unprocessed data, I think he has transcriptome there as well. Unprocessed data, whole genome sequences, RNA-seq for the transcriptome. So transcriptome's kind of in both. I'm not entirely sure why it's in both. I don't know. That's one thing. Try to ask Dr. Blank about that if you're able to ask him a question, but let's kind of go through it. I would be comfortable with these. These are examples. He's going to throw one of them on the exam. So if you have time, after all the other stuff you have to know, try to look on it, but you'll be looking at the mutations that we see in the cancer exome, the transcriptome. Maybe this is the transcriptome only of the cancer. Maybe that's what he's asking. He's saying here. You have the whole genome methylation. Why do we care about methylations of cytosines? What are those important for? Open or closed chromatin, right? If I have methylated cytosines, is that gene going to be expressed or not expressed? Not expressed. So we're doing this just not to look at the actual sequence of the gene. We're looking at transactivator domains, and we're looking at the response elements and whether they're open or closed, whether they're methylated or methylated. So the whole DNA, not just looking at the actual gene itself. Copy number variation can be important if I have a bunch of repeated oncoprotein, over and over and over again, I'm gonna make a bunch of that, I'm gonna transcribe a bunch of that protein, therefore a bunch of those effects. MicroRNAs, what do those do again? Aside from giving you guys headaches, they destroy the transcripts, right? So that's a way to bring down the overall level of the protein. And then clinical information. So in this case, we were talking about before, different drugs, what their symptoms are, what drugs were effective, not effective. That's only through processed data through cBioPortal, not the unprocessed data. So you can go to cBioPortal, and basically they look at that. So what they did is they looked at a unique cancer type, and you're able to look at just all of the tumor suppressor proteins, or all the tumor suppressor genes. Once again, what's the HUGO symbol? It's the symbol for the gene. So they looked at, you can go in there, look at a unique data set of just one cancer type, and then say, what, and out of all of these patients that have this disease, which are the possible tumor suppressors that are mutated? And that's literally all of these right here. And that's what he did, that's what they did. So we'll go over MSI and VNTR instability. So basically the whole idea of MSI, it's microsatellite instability. Basically, it's sort of due to strand slippage, where you basically get screw-ups in MMR repair. Once again, just keep in mind, MMR, what is it for? Repair the DNA. If you have strand slippage, you're often going to get what? You're going to get like bulgings, you're going to get the, what's the word you used for the second exam? Yeah, I think you're going to get distortions. Oftentimes, things like MMR can identify that and destroy it. If I lose MMR and I get strand slippage, I then screw up. I can't identify those distortions. They're going to stay in that DNA, and now I basically get added other nucleotides into that sequence. I better hope I added how many? Three. If it's anything else than any multiple of 3, what have I done then? Frameshift. Now everything's screwed up downstream of there. This is often why now it's not remember, it's not always 100%, but in general, if you have MSI, microsatellite instability due to MMR defects, you are typically going to have truncating proteins. Not typically, but you are more likely to get truncating proteins. Why is that? Because you oftentimes get a frame shift, which means that I have a stop codon appear way sooner than it should, and now that entire protein is truncated. It's not one-to-one, but typically high MSI means a higher probability of having a truncated protein. Because you had a frame shift, and that stop codon then appears a lot sooner than it should. That's what's shown here. This is high MSI instability due to the fact that there's MMR defects. That doesn't mean that all of these mutations now, anything that's green or anything that basically that's a color on here, that means it's a mutation in there. This does not mean that they all have to be truncating proteins, just that there's a high probability of them being truncated. There's even some truncated ones in low MSI, so it's not one-to-one, just about probability. So here, for this certain cancer type, we're looking at all these tumor suppressors. If there was initially a screw-up in MMR, oftentimes in these, what does each one of these little boxes represent? That's a patient, that's a person, that's a unique patient. In this case, what we see overall, in the high MSI group, if MMR was defective, we see a lot more mutations, and a lot of those are going to be truncating mutations. Very, very important. So what happened? I lost my MMR, and then what did I lose? These tumor suppressor proteins. What do those do by definition? Suppress tumors. I've lost them. What happened now? I can't stop proliferation. So when the cell wants to divide, or when the cell divides, it may divide again and again and again. Why? I've lost these tumor suppressors in a lot of these patients. Keep in mind, not every patient, this is all one cancer type, but what do we always say? There's different personalities of the same cancer type. So this individual in particular had this one screwed up, but a different individual had that one down there screwed up, right? So keep in mind how this sort of goes going down.
Kevin,
What's up?
on the side, what is the difference between, I don't know if this is relevant for what we need to know, but with the missense mutation on the key, the putative driver and putative passenger?
I have no idea. I know in general driver mutations, those are the ones that are driving you towards making it a cancer type. Whereas if it's a passenger, I think it's a mutation that occurred, but it's my guess. A mutation that occurred, but it didn't cause a change in the phenotype, whereas the other one does. But they're missense. Missense means what? What are missense mutations?
That is causing another...
It's a different amino acid coming in, right? So, yeah, so it's basically that in both cases you're changing the amino acids, but I think in the passenger case, it's a mutation but doesn't have an effect on the phenotype of the cancer